In this talk, we will discuss different aspects of the evolution of the eukaryotic cell. It is estimated that life on Earth began around 3.5 to 4 billions of years ago. The first living organisms that evolved on Earth were the prokaryotes. Pro stands for before, and karyon means nucleus. Thus, prokaryotes are organisms that were present before the nucleus evolved. All prokaryotes are single-cell organisms, and they can be grouped in one of two domains. Some of them are members of the domain of bacteria, while others are members of the domain of archaea. Prokaryotic cells do not have membrane-bound organelles and hence are a lot more simpler than eukaryotic cells, which contain many organelles that are covered by membranes. Eukaryotes are thought to have evolved after prokaryotes. According to the fossil record, we are able to detect single-cell eukaryotes around 2 billion years ago. Now, U stands for true and karyon means nucleus or kernel. Now, nucleus itself means a tiny kernel or a nut. The reason the nucleus was named this way because when we look at a cell and observe the nucleus, it does indeed look like a tiny nut or kernel that is present in the cell. The nucleus is the compartment of the cell where the DNA is present. In the case of all eukaryotes, we do observe the presence of the nucleus, which is the home of its genetic information, or DNA. All eukaryotes are grouped in the domain of eukarya. There is immense diversity among eukaryotes. Some are unicellular, while others are multicellular. There are 4 traditional kingdoms in the domain of eukarya. The first one is Protista. The organisms that belong to this kingdom that has now been abandoned are protists. The reason it has been abandoned is because the members are genetically very variable from one another, and hence this kingdom is not a monophyletic group. Fungi is another kingdom of eukarya, followed by Plantae, which includes organisms that are photoautotrophs. Animalia is the last or 4th traditional kingdom that is observed in the domain of eukarya. Eukaryotic cells are generally larger in size compared to prokaryotic cells and viruses. There is an exception to this rule. For example, we do have some organisms that are comparable in size to prokaryotes. For example, Austriococcus tauri is considered to be the smallest eukaryotic cell, and it has a diameter of close to 1 micrometer. Eukaryotic cells have membrane-bound organelles, which are usually not present in most prokaryotic cells. The chromosomal DNA of eukaryotic cells is linear, which is different than bacterial or archaeal chromosomes, which are circular. Eukaryotic cells have more complex genomes. The genomes are usually larger in size than prokaryotic cells, and there may be many instances where the cells can have more than one chromosome. For example, in the case of humans who are eukaryotes, every cell has 46 chromosomes, or 23 pairs. Unlike most prokaryotic cells, eukaryotic cells have compartments within the cell called as organelles. Some of these organelles are membrane-bound, like the nucleus, the endoplasmic reticulum, mitochondria, Golgi complex, etc. There are other types of organelles that don't have membranes associated with them, which include the centriole and ribosomes. The different organelles have various functions associated with them, which are different from one another. For example, the nucleus is a membrane-bound organelle that houses the DNA, or the genetic information, of the cell. Thus, it can be considered as the command center of the cell. The endoplasmic reticulum is also a membrane-bound organelle and comes in 2 flavors. The rough endoplasmic reticulum is a site where ribosomes can attach from the cytoplasm, and the ribosomes then translate proteins within the rough endoplasmic reticulum. The smooth endoplasmic reticulum is the site where lipids and other types of molecules are produced. It is also an area where calcium ions are stored in the cell. The mitochondria is a membrane-bound organelle which uses oxygen to oxidize different organic molecules in order to synthesize ATP, which is an energy source of the cell. The Golgi complex is a membrane-bound organelle which collects, packages, and distributes different molecules that are made in the cell, and these can include proteins. Ribosomes are organelles that do not have any membranes associated with them, and their job is to synthesize proteins. All of these organelles are usually present in all eukaryotic cells. Some eukaryotic cells can have organelles like centrioles, which help in the assembly of microtubules, which play important roles during cell division. Centrioles are usually seen in animal cells but are not observed in plant cells. Similarly, lysosomes are also not observed in plant cells, and lysosomes are membrane-bound organelles that are able to break down different macromolecules. Some eukaryotic cells have a cell wall that provides them with protection, while other eukaryotic cells, like plant cells, have chloroplasts. Both of these, the cell wall and chloroplasts, are not present in all eukaryotic cells, and we observe them only in some. A common feature of all eukaryotes is the presence of a membrane-bound nucleus. So how did this nucleus originate in the first place? The most widely accepted notion is that the eukaryotic nucleus arose from an archaeal cell. There is more support for this notion because scientists have found that members of the Asgard superphylum show the presence of eukaryotic signature proteins. There are other ideas that have been put forward, and one of them proposes that the eukaryotic nucleus came from a bacteria instead. Scientists have found that bacteria that belong to the phylum of planktomycetes show the presence of primitive nuclear membranes. So if we look at the picture, the nucleoid of this organism is actually within a membrane that is inside of the plasma membrane, and thus the nucleoid is actually covered by a membrane. Scientists have also found that there are other members also that show the presence of these types of membranes around the nucleus. They have also found that organisms that belong to the planktomycete phylum also show endocytosis, which is a process of taking in substances into the cell. Another idea that has been proposed is that the eukaryotic nucleus actually came from a giant virus. Mimiviruses are known to be large and have large genomes too. Thus, the idea proposes that these giant viruses infected a prokaryote, took some genes from the prokaryote, and thus evolved to form a eukaryotic nucleus. There is some merit to this idea because scientists have found that some of these large viruses, as shown in the figure here, are able to create membranes around the viral genome when they infect a host cell. Another idea that has been proposed is that an archaeal cell formed an endosymbiotic relationship with a bacterial cell. So in this scenario, the archaeal cell was internalized into the bacterial cell, and they both lived happily. Along with time, the bacterial cell slowly started losing its genetic information, and the archaeal nucleoid that was covered by the membranes of the archaeal cell finally formed the nucleus of that cell. Another idea is the eukaryote first hypothesis. This hypothesis states that actually eukaryotic cells that had nuclei were the first cells to evolve, and prokaryotes are actually eukaryotes that lost a lot of genetic information and cell complexity. Thus, there are many ideas that have been put forth in order to explain the origins of the eukaryotic nucleus. In addition to a nucleus, all eukaryotes have membranes within the cell. It is thought that these membranes arose when the plasma membrane started folding inwards. The infolding of the plasma membrane helped in the formation of different organelles like the endoplasmic reticulum, Golgi complex, etc. Thus, ultimately, through multiple rounds of infolding of the plasma membrane, we were able to form many membrane-bound organelles in the eukaryotic cell. Now, one organelle that is present in all eukaryotes is mitochondria. Now, there are very few organisms that are eukaryotes and actually lack mitochondria, but they did have mitochondria before and lost it because they found parasitic relationships with other types of organisms and hence obtained energy from those organisms. Now, in order to explain how mitochondria evolved in eukaryotes, the most widely accepted theory is the endosymbiont theory. According to the endosymbiont theory, there was an ancestral anaerobic prokaryote, and it engulfed an aerobic bacterium that was a purple non-sulfur bacteria that had the ability to take oxygen and use that to oxidize its organic compounds. Genetic analysis has revealed that this bacteria actually belongs to the alpha proteobacterium family. Now, as time passed, this aerobic bacterium lost its ability to live independently and formed the mitochondria within that host cell. The host cell, which is now a eukaryotic cell, was able to adapt to aerobic life because the mitochondria allowed it to use oxygen in order to oxidize its organic compounds. While all eukaryotes have mitochondria, only some eukaryotes have plastids like chloroplasts. In order to explain the origins of chloroplasts, we once again turn to the endosymbiont theory. In this scenario, there was an ancestral eukaryote that was a heterotroph and it already had mitochondria. It was able to internalize a free-living autotrophic photosynthetic bacteria which belonged to the family of cyanobacteria. As time passed, the photosynthetic cyanobacteria lost the ability to live independently and formed a plastid like a chloroplast within that host cell. And thus, we now ended up with a host cell that was photoautotrophic because it was able to use the plastid in order to perform photosynthesis and build its own carbon compounds using inorganic carbon dioxide. There are many lines of evidence in support of the endosymbiont theory. Mitochondria and chloroplast both possess circular DNA, which is observed in prokaryotes, including bacteria, but not in eukaryotes. Eukaryotes have linear DNA. Genetic analysis of the DNA has been performed, and it shows similarity to bacterial members. The mitochondrial DNA shows similarity to alpha proteobacterium members, while the chloroplast sequence shows similarity to cyanobacteria. Mitochondria and chloroplasts both have 70S ribosomes, which are not observed in eukaryotes. Eukaryotes have 80S ribosomes, while prokaryotes have 70S ribosomes. The ribosomal RNA in mitochondria and chloroplasts is similar to bacterial ribosomal RNA. Mitochondria and chloroplasts can reproduce by binary fission, which is widely observed in bacteria. Thus, there are many lines of evidence that support the endosymbiont theory. By analyzing the DNA present in mitochondria and chloroplasts, scientists have found that the mitochondria and chloroplasts evolved only once in time. There are different organisms that have chloroplasts, and some of them include the brown, green, and red algae. By evaluating the chloroplasts in these organisms, it was again found that the chloroplasts arose from the same line of cyanobacteria. However, when we look at the brown and the red and green algae and their phylogenetic relationships, they do not share the same recent ancestor. So it begs the question, how do they have similar chloroplasts? An answer to this question could be secondary endosymbiosis. Let us first look at primary endosymbiosis. This occurred when we had that ancestral eukaryotic cell that already had mitochondria, which engulfed the cyanobacteria. When the cyanobacteria was engulfed, that cell then evolved to form red and green algae. Thus, this first endosymbiotic event is called as primary endosymbiosis. Now let us consider the scenario that this red alga, for example, got engulfed by another eukaryotic cell. This second event where endosymbiosis occurred is an example of secondary endosymbiosis. Now, after this organism engulfed that red alga, the nucleus of the red alga was lost after a period of time. However, the chloroplast remained. This eukaryotic cell evolved to form the brown algae. Thus, the brown algae has the chloroplast that was present in the red alga. As shown in the figure, scientists have found that secondary endosymbiosis has occurred many times. For example, we had a situation where a red alga was engulfed by another eukaryotic organism, and it then gave rise to straminopiles and alveolates, which are two different groups of organisms. On the other hand, we had a situation where another eukaryote engulfed a green alga, which then evolved to form a type of euglyinid. We also had another situation where another type of eukaryote was able to engulf a green alga, and ultimately it gave rise to another group of organisms. Thus, we've had different scenarios where red and green alga were engulfed by other eukaryotes and gave rise to many different types of photosynthetic organisms. The ancient eukaryotic cells evolved with time and underwent many different changes. They ultimately gave rise to the different types of cells that we observe today in eukaryotes. These cells then allowed the formation of unicellular and multicellular eukaryotic organisms. With this, we come to the end of our talk, where we learned about the differences between prokaryotes and eukaryotes. We also learned about the features of the eukaryotic cell and the origins of the eukaryotic nucleus. We also learned about the origins of mitochondria and chloroplasts and what is primary and secondary endosymbiosis. Finally, we saw how diversification of eukaryotic cells has occurred with time.